Okay, let's keep going. And this table tells you what ion channels, what segments, and how they happen compared to graded potential with the action potential. The action potential starts from the axon here lock. You need to travel through the whole axon. And the whole axon, they have the voltage-gated sodium channel, voltage-gated potassium channel covered, so they're able to keep going through the whole axon. I'll leave this video to you. And now let's look at the myelinated. So the previous one happens in the unmyelinated axon. We call it continuous conduction. So they have to keep walking through this. And in some axon, especially they designed to travel for long distance, like motor neuron, and you found the axon is myelinated. It's created by the oligodendrocyte in the central nervous system. It's created by the Schwann cell in the peripheral nervous system. Then what happened is, in the whole axon, over 90% of them being insulated. So only have those small 10%, we call the node or rhombia, they are uninsulated. So the first action potential happens in the first node or rhombia. The second one, they can ignore those 90%. The second one, those voltage-gated ion channel open, is in the second node or rhombia. This is like I ask you to jump, so you can jump from here to here to here. So it's called the saltatory conduction. It happens in the myelinated axon. It's pretty important for the axon need to travel for long distance, like motor neuron. So it will speed up the process. So that's continuous conduction. They don't have myelin, they just keep moving. And the whole axon covered by the voltage-gated sodium and voltage-gated potassium channel. So they just keep opening and the action potential keep walking. For the saltatory, over 90% of the axon being covered by myelin. So the first action potential happened in the first node of rhombia. The second one is the second node of rhombia. So they can jump from here to here to here. It's much faster. It's called the saltatory conduction. And this shows you the saltatory conduction. So once you start to travel for long distance, and the saltatory conduction's advantage really show up, it will speed up the process. And if you damage the myelin, very bad effect. Sometimes the signal won't be able to reach the terminal. And that's what happened in the multiple sclerosis. Like the immune system attack the oligodendrocyte, and you affect the signal to send out to the muscle. And this compared the continuous conduction with the saltatory conduction. And the conducting velocity depends on the axon diameter. So think about the highway. The bigger diameter is actually better. So the one with bigger diameter travel for long distance. And also the myelinated axon travel better than the unmyelinated one. So the nerve, those bundle of axons, they need to travel for long distance. They are usually bigger and they are all myelinated. Now the signal reach the terminal, reach this area, you touch another neuron. So that's the place they're gonna release neurotransmitter, and it will require the calcium. So calcium is the signal molecules. It will open the voltage-gated calcium channel, and when it opens, calcium gonna flow in. Once the calcium flow in, it will trigger the vesicle fuse with the cell membrane, and neurotransmitter is released. And this neurotransmitter will bind with the receptor in the postsynaptic neuron. And you will open, most of them are ligand-gated sodium channels, so sodium flow in, and you create the EPSP. Now this neuron's job is done, and it's this neuron's job to generate its own EPSP and generate its own action potential and keep sending signal down to the next neuron. So that's the synapse part. And we call this presynaptic neuron, we call this postsynaptic neuron. So we spent the whole class talk about how this work, but actually in the neuron, it just take a two milliseconds to finish. And each neuron receive hundreds, if not thousands, of this synapse input. So the neuron is pretty busy. They use a lot of ATP as an energy source. So they create a lot of waste particle molecules. They need a lot of oxygen. And if you block the blood flow to the brain, after 3 minutes, this neuron will start to die, and that's stroke. Okay, I'll leave this table to you. And now let's look at some neurotransmitter more frequently being seen. The first one is acetylcholine. You are pretty familiar with this because that's a neurotransmitter used by motor neuron. So it happens in the neuromuscular junction between the neuron and muscle. We call neuromuscular junction. It also is a neurotransmitter used by the autonomic nervous system, and some of them will happens in the brain, in the central nervous system as well. And these neurotransmitter, they don't recycle. Some neurotransmitter will be packed back and used again. Acetylcholine use the enzyme acetylcholine esterase, and it will break it down. So you always produce new acetylcholine. This neurotransmitter called glutamate. Glutamate is a neurotransmitter used for learning and memory. So you learn something, is what happens is in your brain you create a new synapse. So these synapses help you to memorize all the knowledge, all the things you remember. And if you forget, is this synapse disappear. So neurons always are capable of generating new connection, generating new synapse. And they're always capable of removing those old ones if you don't use it for too long. And it will dissociate, so you can go to connect with others and help you to generate a new memory. So you are always capable of generating new memory. If you want to keep this memory, you need to repeat this knowledge again and again. And what happens in the brain in the molecular level is this synapse, it will release glutamate. And glutamate, and you trigger glutamate release, it will make the synapse stronger. So in the neuroscience, they call it neurons, they fire together, they wire together. So what happens is if you repeat this knowledge again and again, they're going to release a lot of glutamate. And what happens is this synapse stay there forever. So you still remember this knowledge. So repetition is the key for learning and memory. And the neurotransmitter is glutamate. Norepinephrine, that's another neurotransmitter mainly used by your autonomic nervous system, especially the sympathetic nervous system. It will affect your mood, your motivation, also alertness, because that's the sympathetic nervous system. GABA is an inhibitory neurotransmitter. So all the previous three we talk about, they are excitatory. And GABA is an inhibitory. So when the GABA being released, it will bind with the chloride channel. So when the chloride channel open, chloride flow in, enzyme become more negative. So it's created EPSP, inhibitory neurotransmitter, inhibitory post-synaptic potential created by the neuron. So it actually will make the neuron less likely to fire. So the neurotransmitter created GABA still pretty important because you don't want your neuron always fire. You also want to have a break to stop this process. So the medicine to trigger the GABA release, we call the sedative medicine. And alcohol is one of them. So sometimes you feel like you are too nervous, you need a drink. And the alcohol will trigger the GABA release. And you feel calm. That's because your neuron fire less. When you are super nervous, your neuron fire more. So the GABA will trigger the chloride channel open and make the inside more negative. It's a big inhibitory neurotransmitter we found in the brain. Okay, that's it.